Our next speaker is Dannemiller from Ohio State University. She's going to talk about microbial growth in the built environment.
Good morning, everyone. It's great to be here today. Thank you so much for inviting me. I'm really excited to talk to you. So a lot of kids when they're growing up think they might want to be an astronaut. When I was growing up, I wasn't quite sure, but I certainly never imagined that I would study moldy dust. But I absolutely love it. We have some exciting things to talk about for you today. I'm going to spend the first part of my talk talking about some of my work in homes, and that's a spoiler alert. That's going to have some important implications for the work we're doing with NASA. So moldy homes have been a problem for a really long time. And in fact, you can find some ancient Hebrew texts that are now part of the Bible and the Torah that mention the word Sarah F. I was talking on the shuttle today about this, but it's mentioned as growing on skin, on garments, and in homes, and it's actually suggested that we take the homes that are covered in this material and destroy them and throw them away outside of town. So it was recognized as a problem 3,000 years before anyone ever saw a microbe underneath a microscope. If you fast forward to today, exposure to mold in homes costs $22.4 billion per year. And it makes up about 16% of the costs associated with allergic rhinitis, acute bronchitis, as well as asthma. And it's usually not as pretty as this piece of artwork over here, but you can imagine the immense problems that we have due to this exposure to mold growth in homes. And it's a problem potentially not only on Earth, but also in space. So Mark Ott spoke earlier about the challenges that we had with the Mir space station, especially towards the end of its life, with the microbial growth that was occurring due to some damp conditions there. Fortunately, we learned some lessons from Mir when the International Space Station was developed, but anytime you have humans inside this confined environment, we inherently produce moisture. We exhale moisture, we sweat, we drink water, and there's inherently going to be moisture around, and so you have to be careful and you can potentially have some problems. So this is a panel, I think it's been shown a couple times now, of fungal growth on the International Space Station that occurred when an area got too damp. So we need to build towards an understanding of when this can happen to ideally prevent this. And in the future, we want to think about improved spacecraft design. So intentional design of the indoor microbiome is not a new idea. Jessica Green and others have written about bio-informed design and thinking about how we can intentionally design the indoor microbiome in a way to support a healthy environment. And it's really important that we do this so we don't expose ourselves to unintended consequences that could happen if we don't understand what microbes are up there and what we're doing. So this needs to be an integral part of any future spacecraft design. So there's a lot that we need to do to get to the point where we can actually do this, but I see 3 steps that we need to take before we can actually define what a healthy indoor microbiome is and build buildings and spacecraft to support this. The first step is we need to first define what healthy is. And I know Jack Gilbert, Rachel Adams, I've also done some work on health and looking at what a healthy indoor microbiome actually means. And there's a lot of people working towards a consensus on this now. So we have some ideas about this. Next, we need to identify what influences that healthy indoor microbiome and how we can potentially control this, how we might need to design or maintain a spacecraft to promote this healthy indoor microbiome. And finally, we need to be able to assess it. We need to be able to measure and see, did we do what we think we did? And is this a healthy indoor microbiome that exists right now? So my talk is divided into 2 parts. I'm going to mostly focus on what influences the indoor microbiome. The first part I'm going to talk about moisture and microbial growth, and the second part I'm going to talk about work on spacecraft. All of you can hang around for hours 4 and 5 of my talk, where I'll talk about defining a healthy indoor microbiome, some work on asthma, and identifying measurement methods. So, come find me after that. We'll keep talking. So we use a lot of DNA-based methods. So a lot of the work that I'm going to talk about today is focused on dust. And we focus a lot on dust because it's such an important source of human exposure. Here on Earth, where we have gravity, we walk around the room, we resuspend dust into the air, and it's a really important source of human exposure. So we'll usually take the dust, extract DNA, and sequence it to get hundreds of thousands of DNA reads per sample. A lot of others have talked about this and identify the microbial communities that are there. So this is largely what we're doing. All right, part one, building materials and associations with moisture. So we know that damp areas in homes are problematic. We have really strong associations in the literature between dampness in housing and negative health effects. And we've done some work studying growth on things like drywall, but there wasn't a lot known previously about what was actually happening in terms of growth in dust. A lot of people assume the indoor environment was too dry for anything to be generally occurring in the dust, that microbes are generally deposited there, but we wanted to better understand the conditions under which you might actually be able to support this microbial growth in the dust itself, because it's such an important source of human exposure. And so we wanted to ask, what about in carpet? The reason we chose carpet is because in the indoor environment, we get much higher resuspension rates from carpet than a solid surface flooring. You can also see in this graph over here that the relative humidity in the carpet can often be higher than that in the indoor environment, and that extra moisture might be enough to support growth. And there's going to be a lot of things that could happen in a damp carpet, things like dust mite growth, indoor chemistry, bacteria, but we're really going to focus here on the fungi and fungal growth that could occur in the indoor environment. So to do this, we wanted to ask, is relative humidity in the air, not condensed water, sufficient to support microbial growth in carpet? So we collected carpet and dust from homes around Ohio. We exposed it to elevated relative humidity conditions, and then we measured the microbial growth in terms of the rate, the function, and the phthalate degradation. And these are 3 studies that I'm going to talk about today. This is a picture of what it looks like before and after we collect carpet from a home. We selected homes without known moisture problems. I'm thinking about starting a renovation company with these before and after pictures. So if anybody would like a rectangle taken out of your living room, let me know. So we cut the carpet into 10 by 10 centimeter carpet coupons, add the dust, and we embed it with a modified ASTM method. ASTM does have a method for just about everything, including embedding dust in carpet. Works very well. And then we incubate the carpet in a relative humidity and temperature controlled chamber and measure the microbial growth. So first a note about moisture. So when we measure moisture, we're going to be interested in measuring water activity. And water activity is in equilibrium with the relative humidity in the chamber. So water activity of 0.95 is in equilibrium with a relative humidity in the air of 95%. And initially when we started this, we saw that the dust equilibrates very quickly with the relative humidity in the chamber. And in fact, after 1 day and 1 week, you can see visual changes in the darkness of the dust. And after 1 week at 100% relative humidity, we even saw visible fungal growth on the dust. So we knew we were probably onto something here. And indeed, microbial growth can be supported in dust at elevated relative humidity levels without condensed water. So in this graph, we have fungal or bacterial concentrations on the y-axis and the original dust and different relative humidities in the chamber on the x-axis. And you can see that fungal growth starts at around 80% relative humidity levels. And at 100% relative humidity, after 1 week, we have about 27.5 times as much fungi in the dust as compared to what was in the original dust. We do see some growth in bacteria in the red, but only at 100% relative humidity do we actually see an increase in concentration occurring. So the story here is really all about the fungi, which makes sense because it's a fairly dry environment. Fungi can better withstand the lack of moisture. We continued these experiments over about 6 weeks at 100%, 85% relative humidity. And you can see that we had to stop the experiment at 100% after about 3 weeks because the carpet was so covered in visible fungal growth that we thought we had made our point and we could end it there. And we do see some potential growth in bacteria after extended periods of time. What we were also able to determine is that moisture is really the limiting factor for growth in the indoor environment, at least in this house dust that we saw. So we measured various nutrients that might be required for the growth, and we found that in generally they are very much in excess of what you would actually need to support this extensive microbial growth at 100% relative humidity for 1 week. So none of these nutrients were limiting. It was really the moisture that was essentially limiting this growth. And also in homes, the growth in this dust can contribute substantially to human exposure. So we see on top is the fungi and the bottom is bacteria. The orange is the background exposure that you would expect to have in a typical indoor environment. And then this is a rough model of the increased concentration in the air of fungi and bacteria that you might be exposed to if your home had sat at these elevated relative humidity levels. And I should also note that the fungi that tend to grow at these elevated moisture conditions are the ones that contain known allergens. So these are potentially harmful, and this growth in the dust itself can contribute substantially to human exposure. We also wanted to take some pictures. So I don't know if you want to look at this slide. Once you see it, you can't unsee it. But this is fungal growth on carpet fibers at different relative humidity levels. So we see at 50%, 85, 90, 95%, and 100%, you can see an increase in fungal growth on these carpet fibers as the relative humidity increases. So at 80%, we're starting to see hyphal extension, and then at 95 and 100%, we're starting to see some of the fungal fruiting bodies showing up in the carpet, which mimics what we were measuring in terms of total concentration as well. But we can actually visually see what this looks like under the SEM. And so once again, these are the nylon carpet fibers, like this, and then the fungi, you can see examples of that over there. We also took a confocal Z-stack so we could see how the hyphae were attaching to the fibers here, and you can see the nylon carpet fiber in gray and the hyphae extending off of that fiber in blue. So you can visually see what this potentially looks like on these carpet fibers at these elevated relative humidity levels. So this was a really interesting first study, but we realized we wanted to make this more realistic to the indoor environment. So nobody sets the relative humidity in their home at exactly 90% for a week, or at least I hope you don't. But a lot of times the relative humidity in a home is going to tend to have some diurnal variation in it. And it might, the relative humidity levels that we looked at are fairly elevated. So 80% relative humidity is something that would generally only occur under a specific event. So if you have someone showering, or if you have a cooking event, or potentially a hot, humid summer day like here in Houston, and you have the windows open or something like that, or potential water damage. But oftentimes when you have these elevated relative humidity events in a home, they're going to be for shorter periods of time. So someone takes a shower, the relative humidity goes up, and then that extends for some period of time where you have moisture on the surfaces for a while, and then goes back down. So we wanted to understand, can we actually calculate what this growth would be, what the expected growth would be, if the relative humidity varies? So to do this, we collected carpet and dust from homes, 19 homes around the state of Ohio, and we exposed it to variations in relative humidity. So we went from 50% either to 85 or 100% for 0, 6, 12, 18, or 24 hours per day. And we wanted to model this with the time of wetness concept, which was developed for fungal growth on drywall. And what this concept says is that the time of wetness, or the percent of time that the fungi is above a critical moisture growth threshold, is equivalent to the relative growth rate, R over K. And we have the standard model here, and then you also have an activation limited growth model and a deactivation limited growth model up top. So we wanted to apply this to our data. And indeed, we found that we can generally model fungal growth in carpet during these shorter bursts of elevated relative humidity in dust. So here are the graphs. We have the time of wetness, the percent of time that the carpet was above this elevated relative humidity threshold on the x-axis, relative growth rate on the y-axis, and we see it fits somewhat to the standard model, the orange line in the middle, but it is closer to the activation limited growth model, meaning that there is a delay upon the introduction of moisture before the growth actually initiates. But this allows us to actually model this growth in dust and identify under a given set of moisture conditions how much growth would we typically expect based on the relative humidity history in a given environment. So we also wanted to look and see what the difference was between the site effect and the moisture effect. So this is a principal coordinate plot where similar microbial communities cluster together. And the different sites are different colors, and the darker the site, the more moisture that was applied. And can anybody really see the different clusters here? Because what we were expecting is a cluster of, not damp homes over here and a cluster of damp homes over here. And if you squint at this graph really hard, you can kind of see it. Up here, there are some sites that experienced additional dampness, and they did cluster a little bit. But the interesting thing is that the site effects here were really strong. And so it's hard to actually pick out which sites experience this dampness just based on species alone. So we thought that that was really interesting. All right, so now we wanted to ask, what are these microbes actually doing? I really enjoyed Janet's talk talking about a lot of these tools that we can use. So we used a meta-transcriptomic analysis to look at the actual microbial function. And so we looked at samples of dust incubated at 50%, 85%, and 100% relative humidity and looked at the actual microbial function. So this is a principal component plot where similar samples cluster together with similar microbial function. And what we noticed, first of all, is that we were able to obtain RNA from a sample incubated at 50% relative humidity, which was actually surprising to us. This was originally included as a control sample. And so these communities were not actually growing, but there was still active metabolism happening. We got high quality RNA. The data made sense. Lipid metabolism, carbohydrate metabolism was complete. And so you can actually get RNA out of some of these communities, which was really interesting. So the 50% and the 85% were pretty similar, though, in terms of the function of what they were actually doing. But then as you add moisture, you get all of this additional metabolic diversity. And the interesting thing is that the samples at 100% relative humidity are different not only from the 85 and the 100% samples, but they're also very different from each other. So even though these were from the same site, same dust, different replicates, we see that these communities tend to do different things. So this is my favorite GIF. It's going to go viral on Facebook pretty soon, I'm pretty sure. So look for it in your newsfeed. But when we looked at the microbial function, so you can see up in the top left, 50%, 85%, and 100% relative humidity, we saw that at 50% relative humidity, we had a lot of the core metabolic processes in place, the primary metabolic processes, so the lipid metabolism, the carbohydrate metabolism, things like that. At 85%, you get a little bit more around the outside, but at 100%, all of these secondary metabolic pathways around the outside start to light up. And the really interesting thing about that is that a lot of these secondary metabolic pathways are the ones that contain things like allergens and mycotoxins and virulence factors. So these communities that had been incubated at these elevated relative humidity conditions, we hypothesized may tend to produce more of these things. So we measured the allergens. This is all the known allergens that were expressed, comparing 100% to 50%, 100% to 85, and 85% to 50%. And indeed, we did see an increase in the allergen expression at 100% compared to these other two levels. I don't know if I want to say statistically significant after what we talked about, but yes, we did see an increase. And then we wanted to say, can we confirm this with a protein-based analysis? So for this analysis, we had to use a single organism instead of looking at the whole community. But in Aspergillus fumigatus, we were able to show that at increased water activity, we did have an increased allergenicity per spore based on pooled human serum. So we were able to confirm that we have this increased allergen production. So this really asked the question of, is a spore? If you measure a spore in the indoor environment, is it the same, or does it really depend on its previous history, whether it was grown at a lower moisture condition or a higher moisture condition, and is maybe producing some of these potentially more harmful factors? So the third part of this story is we wanted to look at microbial function and see how these microbes might interact with chemicals in the indoor environment. And we wanted to focus on phthalates. We chose phthalates because they're plasticizers, they're common in the indoor environment in fairly high levels in our dust. They're also endocrine disrupting compounds of public health importance that can mimic hormones in your body. We also know that these phthalates are degraded by microbes in wastewater and soil systems, and so we wanted to hypothesize, if we have enough moisture available in the indoor environment, can the microbes in our dust degrade these phthalates as well? So we had 2 experiments. We had what we call a natural experiment. I'm not sure if natural is the right word, but we basically looked at the phthalates that were naturally present in the dust sample. We didn't add any. And then we also had a spiked experiment with deuterated DEHP to look at the phthalate degradation that would occur specifically and actually be able to attribute it to microbial versus other chemical processes that could potentially cause phthalate degradation in dust. So in the first experiment, when we looked at these 9 different phthalates, we have these phthalates over here. On the x-axis is time in weeks, and on the y-axis is concentration. And when you look at the low molecular weight phthalates, we didn't really see a whole lot happen. There wasn't much of a change in concentration, not a lot of degradation occurring. However, when we looked at the higher molecular weight phthalates, we did indeed see degradation of these phthalates in the environment. This was actually surprising to us. We had hypothesized it would go the other way, that we would see preferential degradation of the low molecular weight phthalates. So when we first saw this, we weren't sure why, but we did gain some insight into why potentially later. So another spoiler alert. So we chose DEHP at this point to study for further degradation and do the spiking experiment. And we measured this in 4 different sites, dust from 4 different sites. And in the orange, you can see the degradation due only to microbial processes. And in the blue, you can see degradation due only to chemical processes. But we were able to confirm that under elevated relative humidity conditions, you can indeed get phthalate degradation in your dust due to both chemical and microbial processes. So this was really exciting. We were actually able to show this interaction between chemicals and microbes in the indoor environment, and that the microbes were indeed capable of degrading things like phthalates. So before everybody runs out to put your house at 100% relative humidity to degrade the phthalates in your dust, I would caution you, because it turns out that actually the products of this degradation are potentially more harmful than the parent compound. And also mold growth in your house is probably not a great idea either. But so we measured some of these byproducts as well. DEHP goes to MEHP to phthalic acid, and we were indeed able to confirm that these compounds were produced over time in both the sterile conditions and the microbial growth conditions that occurred, and get a little bit more insight into what was going on here. So I mentioned before that we weren't sure why we saw this preferential degradation of the high molecular weight phthalates, and we were a little bit confused about this. But we did a metatranscriptomic analysis on these communities, and we looked at the gene expression associated with known phthalate degradation pathways. And we looked at a lot of different pathways, but the interesting thing is the ones that we did see upregulated under high moisture conditions were the ones that tend to be associated with preferential degradation of these high molecular weight phthalates. So this is just suggestive evidence at this point. A lot of these genes can be associated with other processes that are happening in the cell, but it appears that the microbes are at least have the potential to degrade these high molecular weight phthalates through these processes that we saw upregulated. So once again, just suggestive evidence, but we thought that this was really interesting, and they appear to be capable of it from the metatranscriptomic analysis. So I think this has a lot of important implications if you think about spacecraft. So we saw that elevated moisture is sufficient to support microbial growth in dust. It's really the limiting factor. When you do have this elevated moisture, you can get this growth, starting at what we saw around 80% relative humidity. We also saw that this has important function in dust, so it can actually change the function and what's occurring and produce things like allergens and mycotoxins and virulence factors at conditions where you have this elevated moisture present. We also saw interactions with chemicals in dust, which has important implications. We have such a complicated system here where we have thousands of microbes and hundreds of chemicals at any given environment, and this is just the tip of the iceberg, so to speak, in terms of what could potentially be happening in our dust every day. And this has important implications not only for homes, but the processes that could potentially be occurring on spacecraft as well. There's a lot of other compounds we could look at too. I'm working with Rachel Adams on a collaboration to look at the VOCs in the indoor environment and potentially interactions with microbes. So she might mention that as well. All right, so the second part of my talk, I want to talk about this exciting new project that it just got started in February, so we have some of the early data, but this is from the HUMID project funded by NASA, so it's the Humidity and Microbial Growth in ISS Dust project. And the main idea from this project is to study dust from the International Space Station and basically see what could happen if things were to go wrong. So this is going to be, once again, at elevated relative humidity conditions, not what's currently maintained up there, but what could happen if, say, you have a small failure in a ventilation system or you have an issue in another environment. Let's identify what this would look like if you have elevated moisture that was unintentional in a given environment. So you basically take the dust, add moisture, and then think about potential unintended consequences for astronaut health, planetary protection if we're on our way to Mars, and spacecraft integrity. So this is a joint project. So Marit Meyer at NASA Glenn and John Horak are on this. John was at NASA for a long time and then came to Ohio State. And then my students Ashley Bope and Nick Nastashi. And so you can see actually a visual change in the dust. So this is dust from the International Space Station that we incubated in our lab at 50% or 100% relative humidity. And you can even just visually see that the dust incubated at 100% relative humidity does indeed get darker. So we looked at the original dust. It looks great. We didn't see very many microbes at all, really not much of anything in the original dust, so it looks like we're doing a great job up there. The really interesting thing about this original dust is that it actually looks very similar to carpet. So I've spent years studying carpet on Earth. Little did I know that I was just preparing to study dust from the International Space Station. So it's very linty dust, it's very fluffy. You get some of that out of homes, but it's much more sort of linty than you get. So you can see the fibers from the dust from the International Space Station look pretty similar to the fibers that you would see in a carpet on Earth. When you incubate the dust, under elevated relative humidity conditions, we do indeed see growth. So we can see growth at 85% and 100% relative humidity. So the dust is pretty similar to the dust on Earth. And then if you incubate it under these conditions, you do get this growth. But with this data, we can start to identify what actually happens under these conditions and start to think about things that we might want to look for as potential indicators of, is this happening? Maybe get some early identification of at an early time if you have a problem occurring. And if one does occur, know generally what's going to happen. We were also able to compare the length of time and the relative humidity conditions for this growth. So this is some of our early data, but we have at 50%, 60%, 85, and 100% relative humidity, you can see the fungal growth that occurs. We don't get much at 50 or 60% as you would generally expect, but we do get growth at 85 at day 10 and 100% by day 5. We get growth in the dust from the International Space Station, once again very similar to the growth in the dust on Earth. So we're seeing the same processes that are occurring here. But there's still a lot of questions to answer. This dust was incubated on Earth, not in the same conditions that occur on the International Space Station. So we still can't identify how the particular conditions in terms of the radiation exposure, the CO2 levels, and things like that might potentially impact this growth that would occur on the space station. So in summary, indoor fungal exposures are very important for your health, and it's important that we control moisture. So if you take away anything from this talk today, it should be that controlling moisture is very important to limit growth in a built environment, whether it's a building on Earth or in the International Space Station. We saw that elevated moisture can cause this growth as a limiting factor. It can lead to enhanced production of virulence factors, allergens, and mycotoxins. It can also interact with chemicals in the indoor environment, and that's almost entirely a black box. We're not sure what's going to happen there. And we see similar processes that happen on dust, whether it's collected on Earth or in space. So it's really important that we continue on the International Space Station to control the moisture as is done up there now, and make sure also that we take this data to understand what would happen if there was some unintentional elevated moisture condition that were to occur. So there are so many people I have to thank for this work. Of course, all the funding agencies, NASA, the Alfred P. Sloan Foundation, HUD, EPA, and NIEHS. And I have a fantastic group of students. They are great to work with, always a source of inspiration for this, for all this work. And of course, all of the awesome collaborators who worked on this, as well as colleagues, other students, and the study participants who contributed their dust, and many of them who responded to our flyer in Starbucks and the libraries about, are you getting rid of a carpet? Would you like to donate it to science? So it's a jungle out there. Thank you very much.